Okay, good afternoon. So in the last few classes, we've been discussing brain's fluid compartments. Right, you have this the cerebral circulation, which consists of blood. Then you have CSF. Then you have extracellular space. Then you have intercellular space. And we said all these fluid compartments are in constant exchange with each other. And this exchange is quite important to maintain the extracellular space at the right level, in homeostatic condition. Because different ionic species are constantly maintained at certain standard levels in the ECS, which is necessary for neuronal function, as we will see later in the next topic. So for that to happen, there must be this constant exchange of small molecules and water, right, among these four compartments. So brain is 20% of takes 20% of cardiac output, right? Whereas it by weight it's only 2%. So it's a huge hog of the cardiac output, the blood flow, and the total flow into the brain is controlled at a fairly fixed level. It's regulated at about 50 ml per 100 grams of brain weight per minute over a wide range of arterial pressures. So there's a huge fluctuation in the blood pressure, right? There is so brain tries to control its inflow at a fixed level of this 50 ml per 100 grams per minute. The metabolic rate of the gray matter is about 4 times that of the white matter, right? Where the cell bodies consume more energy than the fibers. And also the number of capillaries is also about 4 times more. So this number keeps fluctuating. Also, in a different context, I've seen a figure which says number of capillaries is about the same number as the number of neurons, but anyway, it's the same order of magnitude. Now, the thing is, the neural activity and blood flow are closely coupled. Because increased neural activity leads to increase in cerebral blood flow, right? So cerebral flow is like, you can think of it as some kind of an echo or a shadow of neural activity, and this property is used as a basis of some of these so-called functional neuroimaging techniques. Because in neuroimaging, basically you are trying to image the neurons, right? And there are static kinds of imaging techniques where you simply look at the structural properties of the brain. For example, if you take an MRI of the brain, it will only give you the structural properties, things like density distribution. Or if you take DTI, diffusion tensor imaging, it will give you the wiring pattern, which is also structural. Whereas if you want to understand brain's activity, right, you need to do the other kinds of neuroimaging techniques, which are functional neuroimaging techniques. One example is functional MRI, fMRI, right, which is dependent on where you are actually looking at cerebral blood flow patterns. You are not looking at neural activity at all. But since cerebral blood flow patterns closely follow neural activity patterns, right, that measurement of blood flow patterns is justified because it is generally taken as a kind of a surrogate of neural activity. Okay, so yeah. So why should we study this? Like I was saying, some of these imaging techniques like PET and BOLD fMRI. BOLD stands for blood oxygen level dependent, right? And fMRI, functional MRI. They use glucose metabolism and blood oxygen levels as indirect markers of neural activity, because when there is increased neural activity, there's increased local blood flow. Since the incoming blood is oxygenated, so there's increased oxygen levels, oxygenated blood. And when there is higher metabolism, then the oxygen in the blood is removed by the cells, by metabolizing cells. There's a subsequent to that, there's a dip in oxygen levels in the deoxygenated blood. Right. So these changes in blood flow, local changes in blood flow, are markers of neural activity or indirect markers of neural activity, but also changes in glucose metabolism. So the thing is, so you can think of the cerebral blood flow as some kind of a shadow or a marker of neural activity. But the main problem here is qualitatively the relationship is accepted and it is okay. But if you ask quantitatively, that is by measuring a certain magnitude of vascular response or blood flow response, can I estimate what is the corresponding neural activity quantitatively? No, that is not quite well established because those models are not perfect. That is, the quantitative models that relate neural activity to blood flow pattern, or blood flow activity, is they're still not mature. But qualitatively, this relationship is what justifies, right, is the basis of neuroimaging, functional neuroimaging techniques like PET and fMRI. Okay, so this vascular response neural activity has been known for a long time, right? Because when neurons are active, annotate the shared content. Okay, so when the neurons are active, so they release certain substances which are called vasoactive certain substances. Just 1 second. What is this? Okay, and there are many such substances which are released by active neurons, like hydrogen ions, so just protons, or carbon dioxide. Because when neurons consume energy, they release carbon dioxide, right? They produce carbon dioxide, or release of potassium ions. So when neuron is active, potassium from inside goes out, and sodium from outside goes in. So there's a buildup of potassium in the ECS, in the extracellular space. It means the neurons in the neighborhood are quite active. The neurons also release nitric oxide, which is a gas which can diffuse through neural tissue. And nitric oxide can dilate blood vessels. So neurons which are active, which are firing, can release nitric oxide, which will dilate blood vessels. So that is another way. So and also adenosine. So basically, neurons which are active can release one of these many chemicals, many molecules, which can act on nearby blood vessels and they can dilate them. Then, so when we said the neurons, active neurons can dilate the blood vessels locally, this relationship between neural activity and blood flow activity, blood flow patterns, happens both spatially and temporally in a fairly tightly correlated fashion. One thing is temporally the response is somewhat slow because people have found that this response is order of a second, sometimes in a couple of seconds. In very fast cases, it can be a few hundred milliseconds, but otherwise, generally, the response is quite slow. It's order of 1 or 2 seconds temporarily, and spatially, the order is a few hundred microns. That is, if I have a neuron which is sorry, if I have a neuron here which is active within a radius of about 100 microns, that you will have increased blood flow. So increase blood flow. Now, this relationship between neurons and cerebral blood vessels, right? It is important for obvious reasons because you need to understand brain comprehensively, and so it's also known that when neurons are active, the blood vessels dilate and there is increased blood flow, and that is the basis of this kind of functional neuroimaging. So, to understand functional neuroimaging, you need to understand the relationship between neurons and blood vessels, which is also, by the way, called neurovascular coupling. So this relationship is called neurovascular coupling. Okay, so neurovascular coupling is important to understand to interpret, for example, functional neuroimaging. Okay, so that is from the technology point of view, but even otherwise, there's a more fundamental reason why you need to actually study cerebral blood flow also, even if you want to understand brain and neural function. That is because the mainstream thinking about neurovascular coupling is that it is almost like a unidirectional influence. So this is a neuron, and sorry, this is a blood vessel. So a neuron is active, it will release some chemical, and then that will dilate the blood vessel. So activity here leads to dilation, right? So that is a feed-forward influence. So in neuroimaging, this is what we are worried about, we are concerned about. But the thing is, this influence is not just unidirectional. There is also feedback. When the vessel dilates, it releases glucose and oxygen. Oxygen, you can just think of them as energy, which is taken up by the neuron, and that is the energy which fuels the neural activity. So the dependence between these two systems is mutual. It is bidirectional. It's not that neurons are like masters and blood vessels are like slaves. And normally people think of it as some kind of a master-slave relationship, right? Because there is also feedback. This is a mutual relationship. Now, if that is the case, then you can ask some fundamental questions. Suppose I inject some current or something into a blood vessel, or if I separately, spray some chemical on the blood vessel, which makes it dilate, and that will lead to more energy to the neuron, and that will change its firing pattern. So, which means the blood vessel is telling the neuron how it should fire. So, normally we think of neuron as the prime mover. That is, neuron gets activated first, and then that will just dictate to the blood vessel, what it should do and whether it should dilate or not. But then, in principle, you can think of a situation where if you artificially spray some chemical, a vasodilator, on the blood vessel, that will lead to the local change in neural activity. So therefore, you can think of the blood vessel as an initiator of activity, right? And the neuron as something that is responding to blood vessels signals. So this is quite logical. So the point is basically because there is a mutual dependence, you have to look at the influence in both directions. If that is the case, then we need to really rethink the way we understand neurons and blood vessels, because when you talk about the brain function, we typically only talk about neuronal function. We think of all the other systems in the brain as some kind of a secondary systems, as some kind of sidekicks. They're all there, just to serve the neuron. But moment there is a mutual interdependence, you can't say neuron is more important and blood vessel is less important. So there's a lot to be said about it. We've been for the last 10 years in my lab, we've been trying to develop a theory that says that brain's information processing can be best understood in terms of both neurons and blood vessels jointly working together as equal partners. You can't think of neurons as superior or blood vessels as simply following the commands of neurons passively. I don't think that is the case. So in our view, blood vessels also process information. They do information processing just like neurons, and like neurons, they also respond to stimuli. And there's evidence. So we predicted this in a paper in 2010. And just a few years ago, there was a paper in Nature which says that just like neurons respond to stimuli, blood vessels also respond to stimuli in the brain. So and so on and so forth. Okay, so that's a big subject. I don't want to spend too much time on that because this is a short semester and short course, and so if you want, I can send you some papers on that. Okay, so when we talk about blood flow, the inputs to the brain, which are brought in by the arteries, is oxygen, carbohydrates, the amino acids, fats, and hormones, and vitamins, etc. So brain consumes them and metabolizes them and releases carbon dioxide, ammonia, lactate, and hormones. So this is kind of a gross anatomy of the cerebral circulation or circulatory system of the brain, right? There are two pairs of arteries or four major arteries. One is internal carotid arteries and the vertebral arteries. So these two join to form a slightly larger artery called the basilar artery. Okay, so this joins the blood supply of the internal carotid arteries in a ring. So there is a ring-like structure. So this is the ring. So this is called the circle of Villus. So thing is, circulatory system is normally like a tree, right? You have this largest vessel, which is the aorta coming out of the heart, and this progressively branches out into smaller and smaller branches, right? So at the level of any tissue like the brain, you have very small vessels, right, where the exchange is taking place, and these are the capillaries. These are a few microns in diameter, and this is where the exchange of oxygen, glucose, all that is taking place with the surrounding tissue. After that, the capillaries again combine to form venules, okay, and then they form veins, and then the largest veins are your vena cava, superior and inferior vena cava, which come back to the heart. Okay, so this is your so heart is again here. Now, so you see that there is only progressive branching. There are no loops in this. There's only tree structure. loops are present, but only at this microvasculature level. Because when you go to microvasculature, it's not a tree; it's like a mesh. Okay, so because vessels keep merging. So this is a situation pretty much everywhere in the body, except there's one place in the brain where the arteries, some of these large vessels, form a loop, and this loop is called circle of Villus. If you ask why is it there, that's a difficult question because in biology it's very hard to ask, answer the question why. It's a fact, it is there. One thing is, since it is there, you can say that any kind of a loop will give you a more robust kind of architecture because even if one of the branches is snapped or is damaged, you have a parallel route. So in that way, it is good. But this kind of a circular or a loop structure has emerged because of the high symmetry between the two blood hemispheres, right? So that seems to be a kind of a consequence of that. Again, this picture shows various major arteries. Okay, and one of the problems related to pathological conditions related to cerebral vessels occurs because of damage to vessels, and the two kinds of damage: blockage because of some obstruction in the vessel. This is because of thrombosis, embolism, stenosis. So thrombosis is a blood clot, blocking the flow. Embolism is a blood clot forming in one place and kind of drifting along the blood flow and goes to some other place and blocks vessel in some other place. That is embolism. And in stenosis, basically the vessel is become constricted for whatever reason. So, for example, if you eat too much fatty food, the walls of the vessel might be lined by some kind of a plaque. Okay. So it look like that. So because of that, the vessel lumen is now constricted. So that can lead to clots getting stuck inside that because of the narrow passage and all that. So that is stenosis. So all this is blockage type type damage. And other one is bleeding. So this is called a hemorrhagic stroke. The other right, where there is a bleeding of the vessel, that's called hemorrhagic stroke, and blockage of the vessel, that's called an ischemic stroke. So this is ischemic. Now it's third biggest cause, but more recently it is even upgraded to second biggest cause of death. And like I said, two kinds, occlusive and blockage or hemorrhagic. So ischemia means insufficiency of blood supply, and anoxia is insufficiency of oxygen supply. Okay, so that's all. So we have discussed 4 different fluid compartments, and it is relevant for our purpose because the traditional view is that brain's function is all done by neurons, by the more specifically the electrical and chemical activity of the neurons. Okay, whereas a more integral view of brain would include not only neurons, the electric and chemical interaction among neurons, but also the dynamic local volume changes due to interactions among the fluid compartments and dynamic changes in the glucose metabolism and cerebral blood flow. Among these fluid compartments, I just quickly mentioned kind of in the discussion that there are also these cells called astrocytes, which dilate, which can take up fluid and swell, and that way they can redistribute fluid and so on. So astrocytes, in addition to neurons and vessels, there's another class of cells called glial cells, which are also important. Uh, Glial cells have several subcategories. Okay, maybe I'll come to this later in the next topic, right? So brain function seems to be done by 3 systems working in parallel: neurons, these glial cells, and cerebral vessels, blood vessels. All 3 seem to be working together. And you need a really grand theory which can explain how they all work. I mean, I'm talking about a computational theory. If you really want to understand computationally, we don't even have a grand theory of neurons alone, right? But forget about the other 3. But ultimately, we need a grand theory that combines neurons and glial cells and blood vessels. And that will be the really integral theory of brain function. Okay, so we can stop this topic. Then let me go to the next topic. Any questions on this? So until now we have discussed brain's environment. Right now, let us go into brain proper, and let us get down straight quickly to neurons. So although I said that neurons and glial cells and blood vessels are all important for brain function, the mainstream thinking is that neurons are most important, which is still quite true, right? So we cannot spend too much time on glial cells and blood vessels. So let us try to get into the single neuron function. So what are neurons and how do they work? So like I was just saying a moment ago, there are two major cell types in the brain. In addition to neurons, there are these other cells called glial cells. And neurons are special because they are electrically active cells, right? And they have this arboreal means a tree-like. Arbor is tree, right? So they have this rich tree-like projections or a lot of wire sticking out of them, the cell body. And in a human brain, there are 100 billion neurons, and more recent estimate is 86 billion neurons. This size here refers to cell body size. Size can vary anywhere between 4 microns. A granule cell, which is a very small cell body, is about 4 microns, and a large neuron, like the motor neuron in the spinal cord, right, which sends out axons to the muscle, the skeletal muscle, and fires it. So it does a lot of work. When you want to talk about motor function, you're looking at a lot of high levels of energy. So these cell bodies are somewhat larger. So that's about 100 microns in size. Now, if you want to make sense out of, so first let's pay attention to neurons' shape, right? Then we'll try to look at its function. So if you look at pictures of cells, right? Some random, just taken some bunch of random cells, right? You have bacillus and paramecium. So there's a single cell organisms. Then you have cardiac cells. These are cells found in the heart, right? And these cardiac cells you can see. So the heart cells form like a mesh, but they actually look like the bricks in a wall, right? They look very much like bricks in a wall. So you see the partition between cells. So that is a cell, and that's the nucleus of the cell. So it has a strange shape, okay, but not too fancy, but it's a strange shape. And if you look at the basic cells, if you look at the textbook picture of a cell in your some kind of life sciences 101 course, it will generally be a shapeless blob, right, and with some nucleus in the center. Inside that, you'll find nucleolus, and you will find some mitochondria around it, right, and you will find some uh, Golgi bodies, missile bodies, and etc. Endoplasmic reticulum. These are all the standard stuff, right? So it basically looks like a fluid-filled blob. Doesn't have any distinct shape. This is how a typical cell looks like. And here you are looking at some cells which are the first two are okay, just like an extended blob, elongated blob. This is a slightly interesting shape. Now, skeletal muscle is also basically muscle is like a rope, right? It's supposed to contract and produce tension. So this rope is full of strands. And in biology, everything is made up of cells, right? Even something that doesn't look like a cell at all is actually constituted by cells. So here also the cells connect with each other, right? And then form like a rope, like continuous rope. Excuse me. So even the strands of a skeletal muscle, right? The motor fibers are made up of cells. And if you look at RBCs, the blood cells, the red blood cells, they don't look like blobs, but they look more like pancakes, right? And which are almost like a donut, shaped like a donut or a vada, right? If you look at bone marrow cells, it's slightly interesting because bone marrow cells, it's also kind of shapeless blob, but you have these little wires sticking out of it. Okay, so if you look at other cells, the non-neural cells, the shapes are pretty simple. But let us look at neurons, right, and look at their shapes. So here also you have a cell body. You can see this black dot here. But in addition to the cell body, there's huge amount of wire coming out of it. Now, no other cell in the body has this distinctive feature. I mean, even a bone marrow cell has some wire, some wiry stuff sticking out, but it's nothing like a neuron. Neuron is so different from other cells. So even other cells, neurons, just like other cells, has all this standard paraphernalia. It has a nucleus, a mitochondria, right, endoplasmic reticulum. All this stuff is common to any cell. But a neuron is distinctive simply even in terms of appearance by these lots of wires sticking out. So if somebody asks you, what is so special about brain? Why is brain so intelligent? Right? Why don't we say the pancreas is intelligent or heart is intelligent or gut is intelligent? It sounds like a stupid question, but it's not really that stupid because all these different organs in the body are made up of cells, and those cells have the same kind of standard paraphernalia, but there are slight differences. Right? That difference seems to make all the difference because when it comes to neurons, although they have the same essential elements, this wiry stuff sticking out of it seems to be something very fundamentally important, right? And an organ which is made up of cells like this, which has lots of wires sticking out, seem to be capable of very special functions, which other organs in the body are not capable of, right? So this wire part of neurons are very important. Now, if you look at a simple textbook neuron, right, this is actually a special type of neuron called a pyramidal cell. There are many different kinds of neurons. So in this, if you want to label the parts, here's cell body in the center. So this is called the cell body is general term. In a neuron, that is called a soma. Soma means body, right? So it's called a soma. And you have wires sticking out, and these wires are generally grouped into two categories. And so, although it's not always true, you think of a neuron as like a black box, where input comes from one side and goes out in another side. This is a nice textbook description of how neural signaling works. Now, so there are certain fibers which bring incoming information and certain fibers which take out the outgoing information. And the incoming information is brought by fibers which are called dendrites, which deliver the input to the neuron. And the outgoing information is carried by something called an axon. And on the output side, this is output. On the output side, this axon, usually there is a single axon, where towards the end it branches out into smaller branches, which are called axon collaterals. And at the end of at the tip of the axon, so this is the axon collateral, this small kind of a knob-like protuberance. At this point, it makes a contact with a similar knob-like protuberance of another neuron, right? So this is the axon of one neuron, and this is the dendrite of another neuron, and this structure is called a synapse. This junction point between two neurons. So you can think of the dendrite, this synapse occurring as at some point like this. So this picture shows a more detailed picture of a synapse. So this is the axon of one neuron. Okay, so this is an axon. And this is a dendrite. So this whole structure is a synapse. A synapse, by the way, is an extremely complicated structure, very busy area. Lots of fun stuff going on inside a synapse. And people have just worked on what happens in a synapse for their whole lifetimes. So it is. You have the end of the axon, right, which actually makes a contact with the dendrite. At the end, it becomes slightly swollen, and that swollen part is called a button, or sometimes people prefer the French word bouton. Okay, this is called a bouton, and that makes a contact with the dendrite of another neuron. On the dendritic side also, so if you think of dendrite as a cylinder, right? If I just take a cylinder and make a contact with it, if I put a plate on a cylinder, like a disc on a cylinder, it doesn't have much contact surface, like a common contact surface, right? So whereas it's important to have a common sufficiently large contact surface, because the axon actually releases a chemical which gets picked up by the dendrite, and that's how the other neuron gets a signal from the first neuron. So for that chemical to go fully into the target neuron without spilling out into ECS like that, but this is a synaptic cleft as you have seen before, right? And whatever is released from this neuron, from this button, it can go into synaptic cleft and it can also leak out. We talked about this in the last class. So for this stuff not to leak out too much, you want to have very close contact between these two structures. So therefore, nature has designed them so that this face of the button is flat, and the dendritic side also, the dendrite undergoes a small modification where that modification is called a spine. That's called a dendritic spine. This spine surface which presents itself to the button is also flat. So two flat surfaces come together, so thereby creating a very strong contact between this neuron and this neuron, and that's what happens at a synapse. And this flat kind of surfaces, meeting of flat surfaces, make sure that too much of the chemical which comes from one neuron to another doesn't leak out into the extracellular space. So neurons are electrically active cells, right? As I mentioned before. And so any cell actually, for that matter, right, is has some kind of electrical property. But this was, as you know, has been worked out by, our initial results have come from Luigi Galvani, who was considered to be father of the electrophysiology. So in any cell, there is a voltage inside. I'll call this the membrane voltage because this is the cell membrane. And the external voltage, I'll call this like a ground. Okay, ground, which is equal to 0 volts. And internal voltage with respect to ground is, I'll call it Vm. So since this is a voltage across the membrane, it's called the membrane voltage. So this voltage typically for any cell will be a negative value, order of a few tens of millivolts. Okay, in a neuron, that number is roughly around -70, it can be -60, -80, -90, so that's the range. But it's a negative value. Now, so in almost every cell has this kind of voltage. But in a lot of cells, this voltage is almost a constant value. But neurons are special in the sense this voltage is constant at some times in what is called a resting condition. Resting condition, this is at a constant level. But when the neuron gets into what is called an excited state, so this is a resting state, right? And this is an excited state. And when it is an excited state, this voltage is not a constant value anymore. It shows rapid and sharp fluctuations. Okay, this fluctuation is typically in the form of a spike, a very sharp, rapid increase, followed by a rapid decrease, and return to the baseline. Okay, so this can happen over and over again. These spikes are called action potentials. So as you remember, Hartsfield and Huxley has worked out the underlying physiology of the action potential. What is the ionic basis of this action potential? All that they have worked out. So this is what happens when neuron is excited. So this kind of a rapid high frequency fluctuations of the electrical potential is something characteristic of a neuron. And you can say that brain's special privileged position, that as an organ which is very smart, intelligent, all that has partly to do with this other property of neurons. Neuron has two important properties. One is it has all these wires sticking out, and no other cell in the body has that kind of a rich and arboreal kind of exuberance. And similarly, a neuron is also special in another sense, which is it has electrical potential like any other cell, but its potential can show rapid fluctuations. with which it is capable of sending signals to other cell. No other cell in the body can show such rapid fluctuations. And the frequency of this firing can be go all the way to a few hundred hertz, which is considered very high. When you compare to a computer chip can go up to gigahertz and all, which is obviously very high. So brain in that sense is somewhat slow. The single cells are much slower than your computer processors, but brain makes it up by having lots of processors. I mean, which computer can boast of having 100 billion processors? Right? Yeah, we're not even close. So that is why brain single neurons can be slow, but their network can show high speed activity because network is very large. Now, neural signals are electrical waves. That is okay. So neurons produce these waves. These are sharp voltage spikes, and these spikes travel along the. So when a neuron, right, this is a cell body, and it has all these wires sticking out. It can produce these sharp spikes. So these spikes travel along these fibers like that. Okay, and as electrical waves. So here again, so if you can think of neuron as a some kind of input-output system. So where inputs are received from other neurons, outputs are sent to other neurons. Inputs are received via the dendrites, and then these signals get into the neuron. When we say neuron here, it's a soma. Right at the soma, all the signals are combined, and something happens here. I'll describe that in a minute. Then the signals are generated in the soma, and you have action potentials, which then propagate along the axon. Okay, and then at these kind of branch out into axon collaterals. The action potentials also go along these axon collaterals, and they reach other neurons. And once they reach other neuron, at the synapse, this action potential gets converted into a chemical signal. So that's like releases some molecules, right? And that these molecules again act on the other neuron, right? And on the other side again, this is converted into another electrical signal. So I'll describe all that in detail later. So that's how then this signal is received by the dendrite of the other neuron, and this cycle closes. So if you look at the signaling in a neuron, it has four components. First is signal coming in from other neuron, traveling on the dendrites. That's called signal propagation along the dendrites. The next thing is action potential generation in the cell body, right? So basically all signals are summated or added up inside the neuron, and that leads to the generation of action potential. And the action potential generated propagates along the axon. That is the third component. That is signal propagation along the axon. And once the signal reaches the end of the axon and reaches a synapse, right at the signaling at the synapse, the signal gets converted into the electrical signal gets converted into a chemical signal, and in the synaptic cleft, and chemical signal again gets converted into another electrical signal in the other neuron, and the cycle closes. So this continues again. So what is special about the brain? It has these neurons, right? And what is special about them? There are two things that are really special. One is they are arboreal structures. They have all these wires sticking out. And second, they are capable of rapid electrical signaling. So no other cells in the body are endowed with both of them, and also to the extent that neurons have. So no wonder the brain is different as an organ, right? And so you not only have that kind of very interesting types of cells, and these cells are very large in number in the brain, 100 billion neurons roughly, and each cell has 1,000, 10,000 connections. And one more thing, so thing is even heart cells, for example, form a network and they talk to each other, right? They also have electrical activity. The heart is an electrically active organ, right? It fires at a rate of per second, which is very slow compared to a neuron, but it's still fairly decent. But the one thing that distinguishes brain from other organs is that it is also a network, but these connections can make and break, right? It's a dynamic network. It's not only large and complex, it is dynamic. So these connections can make and break all the time. So it's not a fixed network. It looks brain looks fixed only in appearance, right? Where at a microscopic level, the network structure is constantly changing. I mean, your network structure of your brains before and after this lecture will be quite different. I hope it is positive, but it will be quite different. It is more complex than the entire mobile network of the world, because the entire mobile connections of the world is only 5 billion. Let us say, like, even if I accept that every person has a mobile, we haven't yet gotten there. That still be. And number of contacts per mobile. Let us say, how many friends do you have per mobile? Maybe 100, 200, few hundred basically. So that is still nothing compared to a single brain, if you look at it as a network. So you can have an idea of how complex the network brain is. Next, let us come to glial cells. And 90% of the brain cells are, are glial cells. So actually, which is quite shocking because we thought it's all neurons, or neurons are the heroes doing everything, right? And whereas early estimates put the glial cells at about 10 to 50 times more in number than neurons, but more recent estimates put it at 1:1. But anyway, so this is the order of magnitude that gives you the numbers of glial cells with respect to neurons. They don't carry nerve impulses because neurons have this, right? So if you look at an axon, it has action potential, this sharp spike traveling down. Glial cells don't have action potential, but they do have. So glial cell, let me draw. It also has some fibers, some wires sticking out, not as rich as a neuron. And in a glial cell, it doesn't have sharp electrical changes, but it can show fluctuations in calcium. Okay, so you can have calcium waves. These are low frequency waves, but they are quite important. They can be generated in response to stimuli. Okay, so just because they don't have the same kind of signals, it doesn't mean they're not doing any signal processing. They are, right. And glial cells, there are multiple types of them. There are two kinds of glial cells, and you group them as per the central nervous system and the peripheral nervous system. Right. I hope you remember the difference between these two. So think of the nervous system as being distributed in two places. There is the skull and the vertebral column. Okay, so inside the skull you have the brain, and then you have the spinal cord, and from the spinal out, you have 33 spinal nerves and 12 cranial nerves. So all this stuff inside, okay, right, that is CNS. Just like, you see this caption, right, Intel inside, like that, CNS inside. So all this stuff that is in this bony cage is the CNS. And all this stuff, these wires coming out of the nervous system and coming out of the vertebral and filling the entire body. Innervating, that's the word, innervating the entire body. This is called the peripheral nervous system, or PNS. So there are cells, both CNS and in PNS. In the CNS, there are three categories of glial cells: astrocytes, oligodendrocytes, and microglia. Astrocytes transport nutrients to neurons from blood vessels. So basically, when neurons receive, like I was just saying, that neurons receive energy from the blood vessels in the form of glucose and oxygen. So that actually not directly, but there's also some direct transmission, but also indirectly, good part of it comes via the astrocyte, right? Then there is another endocyte which wraps around, providing this insulation, this myelin sheath I was talking about earlier. So myelin sheath is the one which gives insulation to the axons and increases the conduction velocity. So and that myelin sheath is provided by this special kind of glial cells called the oligodendrocytes. Then there's a third kind of glial cells called microglia, which are good at clearing the debris, neural debris. So because neurons are constantly changing, the wiring patterns are constantly changing. In the process, some wire might die and the neuron might regrow new wire. So thing is, another thing, very important thing about neurons you must remember the neural wiring constantly changes, but the neuron as a whole, once it dies, cannot regenerate. Brain cannot produce new neurons once it is mature brain. Okay, so that is, the neurons cannot regenerate. So once as you become old, you keep losing neurons. So that's it. Once the neuron is gone, it's gone. So the only place in the brain, in adult brain, where neurons can grow, right? They are located inside hippocampus, a part of the brain called hippocampus. Specifically, a part of the hippocampus called dentate gyrus. So this is where you have neurons which can keep getting produced, be born there, produced there, adult brain. So it's called adultogenesis. That is in hippocampus. Otherwise, neurons are non-regenerating. So once they are gone, they're gone. In the peripheral nervous system, there are two kinds of glial cells: the satellite cells. The nerves right here in the peripheral nervous system, and Schwann cells provide the insulation. So here also you have this different kind of glial cells called the Schwann cells, which wrap around the neuron, the axons, right, to provide myelin sheath. So this is how the Schwann cell looks like. This is actually a cross section of an axon. You see this coil, right, going around the axon. So this is the purple thing is actually a cell. I mean, it's actually a sheet. Think of it as some kind of a your cutty roll or something like that, right? Like a wrap that you eat. So this is like a wrap, and this is provided by a Schwann cell, and it has myelin in it, so it's a myelin sheath, and this is what gives you increased conduction velocity. There's a disorder called demyelidating disorder, right? And it's called multiple sclerosis. In these people, there is a degeneration of myelin sheath. Right, it attacks 1 in 700 people, and because of this, the conduction velocities go down. So that impairs their functioning in all domains, motor, cognitive, and so on. So they're not able to completely cure it, right? Because we don't fully understand what are all the factors that induce myelination or how to stop this. So this picture gives you the relationship between neurons and glial cells, especially in the CNS. So this is a neuron, right? The cell body, and this is an astrocyte. You see the astrocyte making contact with the blood vessel on one side and neuron on the other side. So it takes glucose and oxygen from the blood vessel, right? It actually processes it and converts it into glucose into pyruvate, and then it sends that stuff to the neuron. The pyruvate, it converts it into lactate. Lactate is then sent to the neuron, where the neuron converts it into pyruvate, then pyruvate into ATP. It is that ATP which is used to fuel the neural activities. The neural activity needs energy, and neurons don't have too much buffering system, right, to store energy. So it depends on this gliovascular system. That is the whole system that combines the glial cells and the vessels. So neurons depend on this gliovascular system to provide them energy in an in-time basis, right? It is like if it is hungry, it should get energy right now. Okay, otherwise it will starve. It cannot keep energy for a long time. So the vessels and glial cells together form a very intelligent energy delivery system, so that they can deliver energy to the neurons in a kind of a on-demand fashion, at the right time, right place. Okay, so neurons and astrocyte networks, and also form networks. So for a long time, people thought that the glial cells are just support cells. In fact, the word glia comes from glue. They thought these are just like a bunch of gum which sticks neurons together. Okay, but which is very uncharitable thing to say about glial cells, because they do very complex signaling. That is nothing. They are nothing like glue. So later on, people understood, since I think about 90s, people started understanding the importance of astrocytes. And just like neurons can talk to each other using certain chemical signals, Astrocytes also can respond to the same neural signals, right? And astrocyte can, so they can listen to it. And astrocyte, the way they listen to neurons is also quite interesting. If this is a synapse between two neurons, right, an astrocyte can send, this is synapse, an astrocyte can send its end foot, right, making a contact with the synapse. And whatever A neuron is saying to neuron B, this actually looks like an ear, right? So this astrocyte end foot can eavesdrop onto the ongoing conversation between A and B, neurons A and B, right, and listen to what they're saying. And not only that, they can even modulate this signal, right? Whatever A is telling B, the astrocyte signal coming from the astrocyte can modulate that and change that. So astrocytes are deeply integrated with neural communication. They're not anything like glue. And also it's been found that synapses form better in culture in the presence of astrocytes. So you can form neural networks in a dish, right? You can maintain neurons right in a dish, and you can induce them to make connections with each other in the dish. And these connections, these synapses, right, form in the culture, form better when you also put astrocytes in the culture. So it's a very integrated system. Neurons and astrocytes work in tandem to perform brain's computation. This is what we've been saying all along. Okay, it's important to just iterate that at this context. So let us stop there, right? Because so far we discussed about neuron components, and the signaling components and the structural components. I also talked a little bit about glial cells.